Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Today we shall discuss about yeast artificial chromosome. Yeast artificial chromosome is a linear vector that behave like yeast chromosome. And yeast artificial chromosome contain several functional modules. One of the functional module is ARS. ARS means autonomous replicating sequence. This autonomous replicating sequence is necessary for replication inside the yeast. Second functional module is CEN4 sequence. This is the CEN4 sequence. CEN4 sequence is meant for centromeric function. The sequence is meant for centromeric function. This yeast artificial chromosome also contain telomeric sequence. This is a telomeric sequence and this is another telomeric sequences. These two telomeric sequences are useful for protection of this yeast artificial chromosome from exonuclease. Other functional module of yeast artificial chromosome is one or two selectable marker. This yeast artificial chromosome known as PYSC3. This PYSC3 contain two selectable markers. One selectable marker is TRP1. Other selectable marker is URA3. This is a selectable marker and this is another selectable marker. This yeast artificial chromosome also contain another selectable marker, that is SUP4. This SUP4 is another selectable marker that is present in this yeast artificial chromosome. This yeast artificial chromosome also contain necessary sequences from E. coli plasmid for selection and propagation within the E. coli cell. Actually, the yeast artificial chromosome is a shuttle vector. That means the yeast artificial chromosome is itself propagated inside bacteria and cloning is done inside yeast. So this yeast artificial chromosome is known as a shuttle vector. The telomeric sequence of the yeast artificial chromosome, these are the two telomeric sequences. These two telomeric sequences are meant to protect this artificial chromosome from exonuclease. The telomeric sequence of yeast chromosome is a 20 to 70 tandem repeat of 6 base sequence that has a hairpin loop at the terminus. So this yeast artificial chromosome is essentially a PBR322 in which the following sequences from yeast chromosome has been integrated. PYSC3 is itself propagated within E. coli, whereas cloning is done in yeast. For cloning, PYSC3 is cleaved by using two different restriction enzymes. One is BAMH1, other is SNAB1. So cleaving this circular vector by BAMH1 cleave this vector molecule at the junction of the telomeric sequence and the fragment that is necessary for circularization within E. coli. This is the fragment that is necessary for circularization of the vector within E. coli, and this is the junction. BAMH1 restriction enzyme cleave this circular vector at these two junctions. After that, this fragment is removed. So after that, this circular vector is transformed into a linear vector. This is the linear yeast artificial chromosome, and this linear vector is digested again by using another restriction enzyme known as SNAB1. SNAB1 recognizes a specific site that is present within SUP4. So after cleaving the sequence between these two segments, the DNA insert is integrated, so a recombinant PYSC3 vector is formed. This is the recombinant PYSC3 vector that carry the DNA insert. After this recombinant PYSC3 vector is introduced into TRP1- and URA3- yeast cells by protoplast transformations. Yeast artificial chromosomes are predominant vector system that is useful for cloning very large DNA segments of eukaryotic chromosome. Friends, this is about yeast artificial chromosome. Please share, like and subscribe this video, and also please give your comments. Bye bye, good night.